Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 3.4, photosynthesis. This is one of the heftiest lessons in the curriculum, cellular respiration being the other one, but we'll be looking at the light-dependent reactions, the light-independent reactions, the components of photosynthesis, the inputs and the outputs, as well as the parts of the chloroplast. And suggested skill is argumentation. 6b, support a claim with evidence from biological principles, concepts, processes, and/or data. First up, a preview of photosynthesis. We have to talk about autotrophs versus heterotrophs. Autotrophs are going to be your producers in the food chain. These are the ones that are going to be making food. Whereas heterotrophs are your consumers and your decomposers. They rely on the autotrophs for both food and oxygen. Because autotrophs consist of photosynthetic organelles known as chloroplasts. And these developed from early photosynthetic organisms and cyanobacteria, which first created the oxygen in the atmosphere and then later joined eukaryotic cells and became the organelle of a chloroplast through endosymbiosis. And like previously mentioned, the chloroplast is going to be the site of photosynthesis in the plant cell. Animal cells do not have these. The equation for photosynthesis is going to be carbon dioxide plus water plus the addition of light energy from the sun is going to be then transferred into glucose and the creation of oxygen. And this reaction can be called a redox reaction or a reduction oxidation reaction, meaning that there will be parts of it, there will be certain reactions that gain electrons and certain reactions that lose electrons. For instance, the carbon is going to be reduced. The carbon is going to have the addition of now oxygen and hydrogen atoms, and with each proton that these atoms bring, there will also be an electron along with them. Whereas the water is going to be oxidated, it is going to be losing its hydrogens, meaning it's losing those electrons that come along with the hydrogens. Now let's take a look at the chloroplast in more detail. The inner gel is known as the stroma, and within the stroma we have thylakoids, membrane sacs. And the membranes of the thylakoids are going to be used to establish concentration gradients, just like from Unit 2. So as you can see, each one of these little coin discs is known as a thylakoid, and a stack of thylakoids is known as a granum. Now chlorophyll is going to be a pigment inside the thylakoids that absorbs light. And it's going to absorb many wavelengths of light, but it does reflect green, which is why plants tend to be green. And stomata are the holes in the leaf through which carbon dioxide enters and oxygen and water vapor leave. So these are going to be the pores that allow for gases to pass in and out of the plant cells, allowing photosynthesis to happen. Photosynthesis is split into two parts. Light-dependent reactions, which, as you may have guessed, are dependent on light, and light-independent reactions, which don't necessarily need light to work, but rely on the products created in the light-dependent reactions. So, the light-dependent reactions are all about producing the chemical energy in the form of ATP that is then going to be used to create glucose in the light-independent reactions. And they're going to convert the solar energy from the sun into chemical energy of ATP. So light is made up of photons, and it's going to be absorbed by the pigments such as chlorophyll within the thylakoids. And there are these different spectrums called action and absorption spectrums that are going to show the efficiency of different light wavelengths and pigments for photosynthesis. And essentially, chlorophyll is going to be best at absorbing red and blue light and will reflect green light. And plants are also able to use more energy from red and blue light for photosynthesis. And the photons absorbed in the thylakoid membranes through groups of pigment molecules called photosystems. And these pigment molecules include chlorophyll, like previously mentioned, and carotenoids, which go for more orange shades. And we have to look at two specific photosystems today. The first one will be photosystem 2. Do not let this confuse you. The order of the photosystems is always going to go 2 to 1 in order that they were discovered. But photosynthesis starts with photosystem 2. Let's take a look now. So, photosystem 2, otherwise known as PS2. begins with a light harvesting compound. And this is a group of pigments that when light is going to be absorbed, one electron is going to be raised to a higher energy state. It is going to become quote-unquote excited. And this energy from the electron is then transferred to the reaction center of the photosystem and later to the primary electron acceptor. So this whole process starts in photosystem 2, which has a primarily pigment of P680. That is the wavelength that it can absorb the best. And these electrons that are harvested in the electron acceptor are going to be donated to the electron transport chain. So let's take a look here. Light energy is going to hit the photosystem, which is going to cause one electron to raise from a higher energy state. It's going to jump around the light harvesting compound until it reaches the reaction center, and then will be transferred to the primary electron acceptor, where it will then go into an electron transport chain, moving on through the process of photosynthesis. However, this process is obviously going to use up electrons that have been taken up by the primary electron acceptor and transferred on. And these replacement electrons are gotten from breaking apart a water molecule. So the oxygen molecules that are released from the breaking apart of water are then going to be released into the atmosphere, which is how photosynthesis produces air that we breathe, but produces the oxygen that is necessary. While the hydrogen ions, because of course we're not using up the hydrogen molecules, just the electrons to replace, the hydrogen ions are going to enter the thylakoid space on the other side of the thylakoid membrane. And now we have to take a look at where these electrons that were harvested in photosystem 2 continue on to. These electrons go from photosystem 2 to photosystem 1. And they do this through the electron transport chain, which consists of cytochrome transmembrane proteins as seen right here. The energy the passage of the electron creates is used to pump protons into the thylakoid space. Essentially, as the electrons pass through the transport chain, this energy generated through the passing of the electrons is used to then pump protons or hydrogen ions into the thylakoid space. This is again also where the hydrogen ions from the water molecule go. This creates a gradient in which there's a lot of protons in the space, but they are unable to diffuse across the membrane because they are ions. Remember, ions have a charge. We have a lot of H+ molecules in the space. They have a charge and are not able to cross the membrane without the assistance of a membrane protein. So to move across the membrane, the hydrogen ions are forced to pass through the protein ATP synthase, which attaches a phosphate group to ADP when activated by the passing of the protons, creating ATP, which again is going to be our currency of the cell needed to create glucose. This whole process is called chemiosmosis. So essentially, the electron transport chain plus some of the protons from the electron transport chain, which produces protons, plus some of the protons from the water molecule are going to make it up into the thylakoid space, creating an imbalance where there are going to be lots of protons in the thylakoid space, but not many on the other side of the membrane. And the only way they can diffuse across the membrane to reach dynamic equilibrium is going to be through the protein ATP synthase. So essentially, the electron transport chain powers ATP synthase by causing a gradient of protons to be built up in the space. And once those protons pass through ATP synthase, this then powers the creation of ATP. It's a long process, but we've almost made it. Our final stop in the light-dependent reactions are going to be at photosystem 1. Remember, we started in photosystem 2, where an electron was excited and donated into the electron transport chain, producing ATP, and now that electron has reached photosystem 1. At the same time, PS1 has also been activated. Light energy has hit PS2 and PS1 at the same time. So more electrons have been donated. So PS1 has also donated some electrons that need to be replaced. And these are replaced from the electrons that came down the electron transport chain from PS2. So the electrons in PS2 are replaced from water, and the electrons in PS1 are replaced from the electrons in PS2. It's a bit confusing, but we'll take a look at the big picture in a second. These donated electrons from PS1 go through the same process of the light harvesting compound, eventually making it to the primary electron acceptor, and they travel along another electron transport chain until they are transmitted to the electron carrier NADP+. This is important, which is then reduced to NADPH once those electrons have been added to it. And the reason this is important is because NADPH is going to be the other product from the light-dependent reactions that is necessary for the production of glucose. And this process is also going to remove hydrogen ions from the stroma, helping increase the proton gradient in the thylakoid space. Because to create NADPH from NADP+, electrons need to be added, but so do some protons. So that's going to create an even larger imbalance between the amount of protons in the thylakoid space and in the stroma, causing them to want even further to move through ATP synthase and create a balance. So now we've crossed the entire light-dependent reactions, and let's take a look at the big picture. We have the total light-dependent reactions. So remember from our total photosynthesis equation, we said that we're going to be using carbon dioxide, water, and light energy to create glucose and oxygen. So we haven't used carbon dioxide yet, but water has been used and so has light energy, and we have created oxygen. So the whole thing, light energy hits both PS2 and PS1, exciting some electrons. These enter the primary electron acceptor and travel down an electron transport chain. In PS2, the electrons that are taken up are then replaced by water, whereas the remainder of the water, the oxygen, leaves out of the cell, creating the air that we breathe, while the hydrogen ions enter the thylakoid space, helping establish a gradient. As these electrons from PS2 transfer along the chain, it's used to pump, the energy from this transfer is used to pump protons into the thylakoid space. When all these protons are pumped, the only way they can make it back across the membrane and diffuse is through the membrane protein ATP synthase, which is then going to create ATP once the hydrogen ions pass through it. So this is the portion in the light-dependent reactions where ATP is created. Lastly, this electron from PS2 is going to replace the one that was taken from PS1, and the electron from PS1 is going to join with NADP+ to form the electron carrier NADPH, which will also be used in the light-independent reactions. So there you have it, the total light-dependent reactions. Now it's time to go on to the second half of photosynthesis, the light-independent reactions. So, light-independent reactions. This is the part of photosynthesis that actually produces the glucose, the food. These reactions do not rely on solar energy, but require the ATP, the chemical energy, and the NADPH from the light-dependent reactions, as well as carbon dioxide from the outside environment, taking our last reactant from the overall photosynthesis equation. 3 carbon dioxides are going to enter the Calvin cycle, and they are going to be incorporated into an organic compound to a 5-carbon sugar through the use of the enzyme Rubisco. Essentially what happens is the CO2 is going to bind to the 5-carbon sugar RUBP, making it an organic compound through the use of the enzyme Rubisco. Now the ATP phosphorylates the compound, meaning it's going to add a phosphate to this compound that's been created, and then it's later going to be reduced, electrons are going to be added through the electron carrier NADPH. Later, the ADP that's left over, as well as the NADP+, are going to re-enter the light-dependent reactions so that they can be reused. So essentially, carbon dioxide is bound to a compound with Rubisco, and then the compound is changed through the chemical energy and the electron carrier that we got from the light-dependent reactions. This is eventually going to form G3P, 5 molecules of it. One of them is going to leave the cycle, while the other 5 are rearranged and reused in the cycle, turned back into RUBP. once two of them leave the cycle, one molecule of glucose is going to be formed. And to create one molecule of glucose, there must be 6 turns of the cycle in order to fix 6 molecules of CO2. And once 3 molecules of CO2 have been fixed, you get 1 G3P. And once the other 3 have been fixed, you get 2 G3P. And ultimately, you get a 6-carbon compound, which is glucose. Let's take a look at a diagram. So, the Calvin cycle is going to use CO2 from the outside environment, the NADPH and ATP from the light-dependent reactions, and it's going to produce glucose, which is food energy, NADP+, and ADP, and these guys are going to go back into the light-dependent reactions to be reused. And glucose is going to be energy stored as food, which is then used by both the plant and consumers of the plant, the heterotrophs, in order to gain energy for themselves. So as you can see here, we have the Calvin cycle. We have 3 carbon dioxides enter and are fixed by Rubisco. They join the RUBP molecule, and once they've been phosphorylated through the ATP and electrons have been added through NADPH, they eventually turn into a G3P molecule, which then leaves the cycle. And we're then left back with these 5 G3P molecules that rearrange themselves to turn back into RUBP. Then the second set of 3 carbon dioxide go through the cycle, creating another G3P molecule, and eventually we've got 2 G3P molecules and 1 molecule of glucose that they make up. But remember, to create 1 molecule of glucose, there need to be 6 turns of the cycle so that the 6 carbon dioxides can be fixed. Although only 2 G3P need to be produced. So, photosynthesis review. Gotta know the parts of a chloroplast, especially the thylakoids and their membranes, which are so important for the light-dependent reactions, as well as the stroma, which is where the Calvin cycle takes place. The photosynthesis equation is going to be carbon dioxide plus water plus light energy yields glucose and oxygen. The water and the light energy are used to create ATP, and oxygen comes from the splitting of the water atoms. And besides ATP, in the light-dependent reactions, NADPH is also formed. This ATP and NADPH is going to then be used in the Calvin cycle in the light-independent reactions along with carbon dioxide in order to fix the carbon dioxide and produce glucose. So, this was a really long video, a really long subunit, but we have covered photosynthesis, and next up we have cellular respiration.